Isis Thesis, Chapter 11, SOS Repair. Why the monster serpent cannot shipwreck the sun god and his crew. Damage to DNA produces single-stranded gaps, but cells have options when cell death is signaled. When extensive UV damage, the cell's last chance to live is survival by the SOS response, which allows a replication fork to proceed past damage. Thus, the cell does not die because replication is not blocked. The drawback to this process is that the 11th hour attempt to save the cell is highly mutagenic. Missing sequences cause unreliable templates that has been referred to as the sinking ship motif, increasing the possibility of mutations. In line with this idea, it may be that the boat which capsized signifies the sinking ship motif, whereas the sunbark, captained by the deceased, proceeds, depicting the reliable template. Deceased king is a magician, a savior. Induction of the SOS response de-represses at least 42 genes that function in DNA repair, recombination, mutation, translesion, DNA synthesis, and prevention of cell divisions. These genes are usually depressed by the transcriptional repressor LEX-A. The inducing signal that de-represses these 42 genes is single-stranded DNA, which binds to RECA protein, activating a cold protease activity. Our model supports that the mobile elements of the deceased inserts into an incoming lambda genome. Our model supports the mobile elements of the deceased insert into an incoming lambda genome, which invades the host cell. During the transcription journey through the cell, the serpent Meehan surrounds the deceased sun god, just as RECA forms a helical filament around ssDNA. RECA protein is a well-conserved multifunctional enzyme with two roles. It not only mediates strand exchange during recombination, but it also can act as a protease, destroying the LEX-A repressor protein during an SOS response. In its first role, it catalyzes DNA strand exchange for recombinational repair and homologous genetic recombination. A major domain binds ADP and probably single and double-stranded DNA. The REC-A protein also has two smaller domains, stabilizing a 6 helical polymer of protein subunits and interhelical bundles, which is believed to be the active biological form, a critical enzyme in the process of homologous recombination. REC-A catalyzes the pairing of ssDNA with complementary regions of dsDNA. REC-A forms a helical filament around ssDNA, extends the ssDNA, binding to dsDNA, which partially unwinds to allow base pairing between the ssDNA and the dsDNA. The dsDNA further unwinds to allow the REC-A driven branch migration. Along with this role, the ssDNA REC-A filament cleaves the LEX-A regulon, de-repressing 42 genes. DNA repair then begins. Now, Let's explore this second role of cleaving or inactivating the LEX-A repressor protein, positing that the monster serpent Apopis is the LEX-A repressor protein. When the recombinant DNA enters the bacterial cell, this penetration along with UV DNA damage signals the SOS response. The ssDNA / REC-A filament is a cold protease activity that cleaves the LEX-A repressor, which catalyzes its own digestion. This results in the depression of 42 genes and the destruction of the dragon, so to speak. So we could say that the deceased restores the whole eye of horse, crow protein production in the genome for the lytic pathway and excision of the prophet.